In this video, we will discuss about a new paper that shows that immunotherapy activated T cells can recruit and activate M1-type macrophages. So, in order to completely eliminate the tumors through immunotherapies, it is essential that both the innate immune response and adaptive immune response should work together. Then only it is possible to get rid of the tumors completely. So in this paper what they have found out is that the activated T cells can recruit and activate this late M1 type macrophages. So we will understand the graphical abstract in this figure, and in the subsequent videos we will go through the experimental results that they have presented in the paper. So we will start with this point where the T cells based immunotherapy or activated T cells is injected into the patients, and then it will cause this T cell influx and activations once they identify the tumors within the patients, and then it will release the cytokines that will lead to the killing of the cancer, and this killing of this cancer will cause release of this further epitopes that can help in the macrophages influx. And this macrophages influx, when they reach to the site of the tumors, so they are getting activated through two different mechanisms. First one is this late stage activations, where these effector cells will the effector cell release IFN, gamma, and other T cell receptors ligands that helps in the activation of this M1 like macrophages. And other things, the macrophages that are in the vicinity of the these activated T cells or T effector cells, they get activated. They activate these monocytes into the M1 type macrophages through this CCR5 interactions. That is, the cytokines that are released by these factor cells will interact with the CCR5 receptors on the monocyte or M0 macrophages. And after that, what will happen is that now the conditions reaches to a point where the tumor cells will be targeted not only by the activated T cells but also by this M1 type macrophages, which are indirectly recruited by the activated T cells and activated by them through the release of the cytokines. And this shown that the blue region shown is that is the minimum, that is when the T cells is injected first, then what will be the response, and when it reaches to the maximum level, the immune response at maximum levels is the combinations of both the innate immune response and the adaptive immune response. And so this is just a model of the macrophages T cells interactions in cancer cells, and they check their results both in the in vitro form or in vitro lab studies and also in the mouse models. And what they have find out is that the M1 type macrophages results transcriptome results of the M1 type macrophages, activated M1 type macrophages, is similar to that of the M1 type macrophages transcriptome results that they have find out from the in vivo studies. So this indicates that the mechanisms that they have presented could actually be replicating in the actual scenario or in the mouse models. This could be one of the possible ways by which these immunotherapy activated T cells can be used along with the macrophages, but simultaneously it will also give us a warning that these M1 type macrophages get all activated. This M1 type macrophages, activated M1 type macrophages, might also has some of the side effects, that it might also affect some of the cells in the vicinity of the tumor cells, because these tumor cells are densely surrounded by the healthy cells, and the M1 type macrophages might find it difficult to distinguish between the healthy cells and the tumor cells. So this is what the idea or the graphical abstract of the paper. If you need to understand more about these papers or their abstract or other experiments in detail, so that you will understand the upcoming videos in a better manner, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.